Hi everyone, I hope you're doing really well. Today we are going to be analyzing some RNA-seq data from DEE2 with the tool Degust. Degust is really nice because you can do tons of different analysis in the cloud. So let's get started. And to do that, we need a data set to begin with. And I've got this really nice data set from Geo. I'll just go to the here it is here. So what we've got is an RNA-seq expression data set, which is AML3 cells, either control or treated with the drug azacitidine. And azacitidine is quite interesting because it's not only used in the clinic, but its mechanism is thought to be on DNA methylation. So it's a good one for us to investigate. So having a look at the samples that we've got in this experiment, we've got treated and untreated samples with 3 replicates each. And what we will do is grab this geoseries number and we'll go over to the DEE2 website and we'll use the search tool. And this is a human data set, so we're going to select human and paste in the geoseries here. And this is what you get when you have just one result. We're going to download this data bundle here. And there it goes. Let's open this with our file explorer. And we can see we've got, that's the zip file that was downloaded. And I think it's already been extracted here. We've got a README. I won't open RStudio just now. What we'll do is open with maybe gedit, just our text analysis tool. And you can see we've got all the different descriptions of all of the contents here, including the gene count matrix that we're going to use in today's analysis. So the gene count matrix is this one here, and I'll open it with our text editor. And you can see we've got the runs across the top and the genes across the side. So that should be good for us to load straight into Degust. Let's give it a try. It's at degust.erc.monash.edu, and we can get started by downloading this and gene count matrix, uploading the gene count matrix. Okay, so we're going to call this one ASA. This is RNA-seq counts, tab separated, and the gene ID is going to be our information column or kind of annotation. And then we add our conditions. The first condition will be control. And I've already collected the mapping information between the sample ID and the treatment group and the SRA run. So I know that control 1 is going to be this one here, 523. So we might actually read some of these samples. So we know that this is control 1, control 2, control 3, ASA 1, ASA 2, and ASA 3. So make sure you check this very carefully. So we'll call this control 1, 2, 3. Done. ASA 1, 2, 3. And if you've just got 2 sample groups here, and you do the control first and then the case, and you hit save changes, you're good to go. and it's going to start doing the processing of data like normalization and differential expression. And by default, it uses the Voom-Limmer method to do this. It's giving us a note here that we haven't set a minimum expression filter. Now, this is not like a universal thing, but I think it is a good idea because genes with very low expression, they tend to be quite noisy. And if we eliminate them because they have no chance of being differentially expressed, then it's going to make our downstream analysis a little bit better. So I'll click on configure and we'll set a minimum read count. And maybe I'll call it, I'll set a minimum gene counts per million of 1 in at least 2 samples. So see what that does. It should cut it down from 58,000 down to 20,000, something along those lines. 13,000, that's actually quite good. So what we can see is we've got in blue genes that are not changing their expression, and in red we've got all of the ones with strongly changing expression. Let's pick this gene here. Yeah, you can see there's a big difference there. We can also change the method of differential expression. This one is EdgeR based analysis. Okay, and that looks pretty good. We can also look at other charts like an MDS plot. We can see that there's a big difference between the sample groups, which is good, and the volcano chart here. So that is general gist of this. I'd recommend you download the CSV file of the results and also download the R code like this and save those for your electronic notebook. And also the data here, the files that you've downloaded from DEE2, you should also save those to your electronic notebook for future reference. So I hope you found this guide interesting and useful, and I hope to see you in the next one.